Yes.
where I'm moving my mouse.
Yes.
So thank you. I feel privileged to be able to come and talk to you about RGD, about the Alliance, about the models and the diseases that we have and that we curate. I am going to barrel on through, so if anyone has any questions, please feel free to interrupt me and ask. And otherwise, let's go. And there we go. Okay, so just to give you an overview of what I'm going to talk to you about today, I'm going to talk about finding disease-related data in RGD. So I'm going to talk about ontology searching and browsing to find strains, and then our RGD find models tool, which is also a strain search tool. And then we're going to look at an example of a genetic model of obesity. We'll look at the strain, the allele, and the variant. And just so we're all on the same page, we define an allele as basically a change in a gene. So it's a gene level object. And then the variant is the actual molecular change that has been made or has been found in that allele that makes it different from the parent gene. So then I'll look at some genes and variants in terms of Rat Lepr, and then I want to talk to you about the RGD curation practices for disease gene curation. Then we'll look at the human Lepr, and we'll talk about the ClinVar variants and disease annotations. We'll take a very, very brief look at JBrowse, basically just touch on that. And then we'll look at genes and variants at the Alliance. So human Lepr, and then the high volume human variants that have been imported there, and also the rat variants that are there. And then I'm going to talk very briefly about RGD's variant visualizer. So RGD is the Rat Genome Database, but we are more than just rats, and we're more than just genomes. RGD is actually a multi-species knowledge base, and we have data for rat, obviously, but also for human, for mouse, for chinchilla, for the domestic dog, for 13-line ground squirrel, bonobo, pig, pig as a biomedical model, not pig as an agricultural animal. And then most recently, we've added the African green monkey and the naked mole rat to our group. So RGD's manual curation is both comprehensive and diverse, very disease and gene focused, although we are also, for rat, we are also focused on strains, and we have done some QTL curation, although we've kind of gotten a little bit away from that. But we do prioritize our curation based on our disease portals, which are basically consolidations of data for a particular disease category. So for instance, we have a cardiovascular disease portal that has information about genes, QTLs, and strains that are associated with cardiovascular disease, and then also across species. So cardiovascular disease genes across all of the species, and a couple of analysis tools that I'm not going to talk about today. But we do cross-species annotations. We do rat and human for both disease and phenotype, and we do limited curation of genes in other species. And then we have multiple data types and multiple ontologies. So we have genes, we have gene alleles, we have QTLs, we have strains. We work with the disease and both mammalian and human phenotype ontologies. And then gene ontology, pathway ontology, we do gene chemical interaction annotations, and then we have quantitative phenotype measurements for rat. And I am not going to talk about the quantitative phenotype measurements. We're actually in the process of overhauling our displays and our tools for using that data. And so it's kind of in a state of flux, but that might be something that could be of interest, because it's not just saying that the blood pressure for a particular rat is increased or decreased. It actually will give you the measurement value for say systolic blood pressure in a hypertensive rat under high salt conditions. And so it's very detailed, and it gives you very good information about actual phenotype measurement values, not just qualitative relative terms. So in terms of disease and phenotype curation for strains at RGD, we have a number of criteria that we use for making strain disease and strain phenotype associations. Strain disease and phenotype annotations are almost always based on differences between genetically diverse strains. Traditionally, that was between non-mutant strains. So there's a whole a large number of inbred rat strains that have been developed as models for human disease. And a lot of those have spontaneous mutations. But more recently, with the rise of zinc finger mutations and CRISPR modifications, experimentally mutated rats are also available. So we do strain disease and phenotype annotations for those. The evidence codes, because they're genetically based, are IMP and IAGP. IMP is inferred from mutant phenotype. Inferred from mutant phenotype is used when it is an experimentally mutated rat, and then IAGP is inferred from the association between a genotype and a phenotype, and those are used for the naturally occurring variants. Curations enter the compared to strain information in the notes field. So if they're making an annotation to a hypertensive rat, and they're saying that the blood pressure is increased in that rat, they will put in the notes field increased compared to what, because it's not necessarily a parent strain of an experimentally mutated or a knockout rat or whatever. Very often they are inbred strains, so you need to have the information about what that is compared to be able to say it's increased or it's decreased. We use qualifiers to extend the information associated with an annotation. So for disease, we use qualifiers such as induced. It's associated with a treatment. This is associated with penetrance. For phenotype, sexual dimorphism and severity are also relatively commonly used. We use the term model in the qualifier to designate that a strain is an established model for a disease or phenotype. And there have to be at least 2 or more publications to support that designation. And model control is a qualifier that's used and assigned to strains that are established as controls for a particular disease or phenotype. So for example, the WKY strain is commonly used as a control for the hypertensive SHR strain in the study of blood pressure. And you have to know that a strain might be a control for one phenotype, but it might be affected for another phenotype. So it's, or disease. So it's important to pay attention to what the actual disease or phenotype is that you're talking about if you're looking at a strain as a control. So in terms of finding disease-related data, you can just put your disease term in our general search, or we have an ontology specific search. And when you do that, you'll come up with genes, strains, QTLs, SSLPs, variants, a bunch of data across species, and a number of different data types, but you'll also come up with this information about how many terms there are in each of the ontologies that we use that match your search criteria. So in this case, we'd be most interested in the human phenotype, the mammalian phenotype, and the disease ontology. So if you choose one of those, it takes you to this ontology result page where you can select which you can see all of them. When you go in, by default, you see all of the ontology terms, but you can select a particular ontology and just see the terms from that ontology. If you click the term or you click this little A icon, you'll go to the actual list of annotations. If you click this leaf icon, it takes you to our ontology browser, which looks like this. It doesn't look like anybody else's browser. So the term that you've selected is shown here in the middle. Its parent terms, one or more parent terms, are shown on the left. Any children of these parent terms will be shown in this center section with the term that you've selected as sibling terms, and then all of the children of this term will show up in the right-hand panel. And here again, if you choose the A icon for any of these terms, you'll go to the ontology report page for that, or in these links up at the top can be used to access the ontology report page with the view of these that you've chosen shown by default. So here I've chosen rat, and this is the ontology report page. You have the information about the actual term itself here at the top, and then there's a display of all of the annotated objects that can be mapped to the genome shown in the genomic context, so in the context of the karyotype. And the brown is genes, the blue are QTL, and the green are strains that can be mapped, so either mutant strains or congenic strains. And then below that, there's the list of annotated objects and the annotations, information about the annotations. The default when you come in is genes, but I've selected strains because that's what we're talking about right now. So this shows the list of 29 strains that have been annotated to the term obesity. And you get information about the symbol, the name if it has one. Here are the qualifiers and the evidence codes, as I was talking about. The notes that say, in this particular case, this F344 strain is used as a control compared to the FDIO rat strain. And then you have links to the references that that information came from, either at PubMed or the reference at RGD, which gives you the link to the full paper, and also will give you a list of all of the objects that have been annotated from that paper and all of the annotations that were made. And then for the mutant and congenic strains that can be mapped to the genome, the actual genome position is given, and a link to JBrowse to be able to see it in the genomic context. So if we choose, no, I'm sorry, got ahead of myself. Okay, so the another way of finding rat strains at RGD is what we call our Find Models tool. This is an ontology-based search tool that is specific for strains. So I've put obesity into into the search box, and if I hit my search go, I get the list of strains that have been annotated to obesity. And you can filter using these facets on the side. You can filter by that qualifier. So if you only want to see the spontaneous models, then you can choose that. Or if you only want to see inbred strains, you can select a strain type. And we also have started with, this is a fairly recent thing for us, started including conditions in our qualitative annotations. That's something that we've used for our quantitative phenotype measurement annotations in the past, but we've recently started using those for our qualitative. So you can see in this case, this rat is an induced model of obesity, and it's induced under the conditions of controlled calorie content diet or controlled fat content diet. So the strain symbols in this column are links to the strain page. So if I click on this link, then I'm taken to this strain page for this actual knockout rat for the Lepr gene. So this gives you a bunch of information about the strain, including, I mean, obviously the strain symbol, the link, the ID at the research identifier ID. We have an ontology of rat strains. Basically, it's rat strain derivation. So it gives you a link to that. It tells you what the allele is, if there is one, which in this case there is. It tells you something about the origin of it, and where position information is available. It gives position information on multiple assemblies. It also, below that, it will have the list of disease annotations that have been made for this particular strain. So in addition to obesity, this strain is a model of hyperphagia, and also shows proteinuria in a sexually dimorphic fashion. And also its sexual dimorphism has been seen for obesity in that particular rat. And you can see that both of those are compared to the BN. And then there's also phenotype annotations. So mammalian, these are from the mammalian phenotype. And so it gives you the whole list of actual phenotypes that have been seen. So not just the diseases, but basically the phenotypes that underlie those diseases, with all of the information that we've talked about. So going back up to the top of the page, if I want to see the allele, information about the allele, this is a link, I can go to the allele page if I follow that. And up until very recently, this description of what the mutation was the only information that we had about the variant for this particular allele. We have more recently started, and we're still in the process of actually creating variant records for our gene level allele records, where that information is available and findable. And so if I'm interested in what the actual variant is for this mutation, I can go to the variant record, and it actually gives me the genomic position of the variant. It shows the reference and the variant nucleotide. It tells me that it's a point mutation, and then it gives me links to the various strains that contain this allele that has this variant. So if I want to go to the if I want to go to the parent gene, if I want more information about the parent gene, in this case Lepr of this allele, I can either go back to the allele record and it gives me a link to the parent, or I can just search for that gene symbol, and I get all of the genes in all of the species that match that gene symbol. And then, as we've seen before, additional data types that match that search criteria. And then if I choose view all, I can choose a specific here, let me go back. Nope, can't do that. Let me go back. Here we go. So if I choose a specific species, then I'll see all of the information, all of the data types that are in that species. If I choose one of these numbers, I'll just see the genes, in this case, the genes in the rat, or I can see the genes in all of the species, or I can view all of the data. And that gives me a list of all of the genes, all of the strains, in the case of human, all of the variants that match that search criteria. And so if I want to go to the gene record, then I would select, in this case, I'm going to select the rat Lepr and go to the gene record. There is a lot of information on this, and I'm not going to have time to go through all of it. But we have ontology annotations. So there's a whole list of ontology annotations. And the gene description is based on the ontology annotations. We have links to the orthologous genes in RGD. We have links to the gene and its orthologs at the Alliance. Links, in this case, to alleles of the gene, and also to the strains, the mutant strains that carry those alleles. We have positions on multiple assemblies, and then disease and phenotype ontology annotations. So you can click one of these and it will take you down the page and show you the disease annotations. But before we go there, I wanted to mention basically how we annotate disease gene associations. So the question is, how is a gene related to disease? We don't only do annotations based on genetic differences. For genes, we also we look at biomarkers for diseases. So if the gene product is associated with the disease by expression level, we will give it an evidence code of inferred from expression pattern. And it doesn't say anything about causality, but it might be a pointer towards something that might be interesting. And that we found that kind of data is much more common than actual genetic modifications. So we actually do these biomarker annotations. We do annotations to molecular mechanisms where a gene product is known to be functionally involved in the disease process, and give it an evidence code of inferred from direct assay. We do annotations for therapeutic targets where the gene product is a target for the drug, which is therapeutic for the disease. This can be an IDA evidence code. Actually, it's more commonly an IMP, so inferred from mutant phenotype evidence code, because very often they're using inhibitors of the gene or the gene product to be able to show that it's a therapeutic target. Or yeah. And then also the obvious, the variant associations, which are usually polymorphisms or mutations. So the gene sequence is being distinguished from others that may be involved in the disease. And again, for genes as well as for strains, the evidence code is IAGP if it's a naturally occurring polymorphism, and IMP if it's an experimentally induced mutation, or for non-mutation mutational variations such as antibodies or RNAi. So given that, I will show you some of the disease annotations for the rat Lepr, and this is only a subset. There are actually quite a few more. And you can see you've got several qualifiers, including no association. In this case, it's this is actually the human gene. So there was a study that was done, and they looked specifically to see if they could find a mutation that was similar to the rat FA, so fatty mutation, and were not able to find it in the cohort that they were looking at. Okay, so where possible, IAGP annotations will give the RSID or another designation for variants. So something like the Q223R designation, but wherever possible, we try and find the RSID. Currently, this is being put in the notes, and they're structured. The format of the notes is structured with an eye toward extracting the information to make it more usable. So to actually be able to pull out the RSIDs and be able to have a field that was searchable, to be able to find these RSIDs and associate it with this gene and non-alcoholic steatohepatitis. So if an association with one disease is found in the context of another disease, this is included in the notes. So type 2 diabetes mellitus in this cohort, it was associated with non-alcoholic fatty liver disease. So all of the members of the cohort had non-alcoholic fatty liver disease, and they found this variant was associated with type 2 diabetes mellitus. So that's what this note is telling you. And you can see here that we have an annotation, an IEP biomarker annotation, and it gives you information about what the molecule was that changed expression or changed activity. So in this case, it's an mRNA. The expression was increased, and it was shown to be increased in the small intestine, and in the context of short bowel syndrome. So we also do annotations. We store data for human and do annotations for human, as I mentioned before. So just to highlight, give you some highlights of the human Lepr gene page. Again, the ontology-based ontology annotation-based gene descriptions, the links to orthologous genes at RGD, and to that gene and its orthologs at the Alliance. Obviously, we do not have alleles or strains for human genes, but we do have ClinVar variants that we have imported. And so there's a link to the ClinVar data, which actually is farther down on the page. Then there's positions on multiple assemblies, which could be of interest to the clinicians, because as we understand it, a lot of the clinicians are still using build 37. And so we still have the build 37 and also build 38 positions, both from NCBI and from Ensembl. And then we've got disease and human phenotype ontology annotations. And then I mentioned the links to the imported ClinVar variants. So RGD imports phenotype annotations from HPO and also curates HPO annotations from the literature. So we've got for Lepr, not surprisingly, there's a whole list of phenotypes that have been associated. Imported annotations show where they were derived from. So we get them from HPO, but they're deriving them from OMIM and also from Orphanet. Excuse me. And then the same type of structured notes are included with RGD's curated annotations to tell you for this one, for these, for instance, the RSID and where it's located. Human phenotype and mammalian phenotype annotations are only done based on genetic differences. We don't do expression-based annotations for phenotype, only genetic. And you can see that using the evidence code there. So RGD imports variant records from ClinVar and matches the conditions that ClinVar lists to DOE terms to actually create disease annotations. So when we get the data from ClinVar, it has, you know, obesity, monogenic non-syndromic obesity, obesity due to leptin receptor gene deficiency, all kinds of relatively controlled but not ontology-based text that tells you about the conditions, and we match those to actual disease ontology terms, and then make disease annotations for both the ClinVar, the variants themselves, and also the associated gene. So in this case, what's in the with field here is the link to the ClinVar variants, and then we've got the in this case the match to diabetes mellitus as the disease term. So genes and variants can be viewed in JBrowse, so you can explore them in the genomic context. So for instance, this variant, this ClinVar variant, you can see it in JBrowse. Here's the Lepr gene, and then you can see this is the variant, this one right here. If you click on this label, it will give you some information about the variant itself, including the trait that it's associated with, whether it's considered pathogenic or not, the molecular consequences, and some other information from ClinVar about the variant. So rat and human gene and variant data is also available at the Alliance of Genome Resources. I understand that you got to get an overview of the Alliance from Carol Bolt and Cindy Smith in the past, so I'm not going to go into great detail, but I do want to show you some things that have to do with the rat and human genes and variants at the Alliance. So this is the human Lepr gene page, and it gives you some general information. You've got the symbol, as you would expect, and synonyms, the automated description. The automated descriptions that we have at RGD are actually derived from the Alliance. We actually download these from the Alliance. So there's an algorithm that was developed at the Alliance that actually looks at the ontology annotations for the gene and the human ortholog, if you're talking about model organism genes. It looks at the human ortholog and then writes a summary of the diseases and functions and processes and subcellular localizations that those genes are associated with. And then it also, we're actually the ones that submit the human data to the Alliance. And so it gives us credit for this description, which is very nice, but this is actually the description that comes from RefSeq. So we include the RefSeq definitions where they exist for genes in our submission to the Alliance. So one of the advantages of the Alliance is the ability to see the orthology across diverse species. In this case, Lepr only has good orthologs in rat, mouse, and zebrafish. I don't know how much was talked about the stringency levels at the Alliance. So the default is to show the stringent level, which is orthologs that are called by at least 3 algorithms, and either are the best or best reverse predicted to be the best or best reverse hits, or 2 orthologs and both best and best reversed. So so these are the stringent orthologs. So you can see at least 3. You can take that down to moderate, and it's not quite as stringent, or you can take all of the filters off and see everything that any of these algorithms have predicted as orthologs. And if you take all of the filters off, in this case, there are actually a couple of worm genes and a couple of fly genes that have been called as orthologs for the human Lepr gene by a single algorithm. So it's not considered the best, but if you are looking for orthologs and you can't find what you're looking for, you can take all of the filters off and see if there are any there. So in addition, the human phenotype annotations have been submitted to the Alliance. We have submitted those to the Alliance. So human phenotype annotations are available. Also, my arrow didn't move. Disease annotations. And one thing that's very nice is this disease ribbon that gives you an overview of the annotations available for each of the species. And the species that are shown here are based on the stringency selection that you've made above. So these are, again, the more stringent of the orthologs. So it's, in addition to human, you're seeing, you can see annotations for mouse, rat, and also zebrafish. And here it's, we have the evidence codes that are assigned. These are all derived from RGD. The evidence codes that are assigned, but that determines what the association is between the gene and the disease. And so it's actually spelled out that in this case, because it's based on expression pattern, Lepr is a marker for arteriosclerosis. But in this case, because it's based on genetics, Lepr is implicated in dilated cardiomyopathy. So almost 360 million human variants have been imported from Ensembl into the Alliance, and those are available on the gene pages. So you have this list, and you can imagine that with 360 billion human variants, there are a lot of them for any particular gene. In this case, for Lepr, there's over 47,000 variants that have been assigned to that gene. So that would be a bit overwhelming, but you can see that there are a number of filters that can be applied. So in this case, if I want to filter down, say I'm not interested in the intronic variants, I'm only interested in the ones, say, that are missense or stop gained or a splice acceptor variant. For instance, I can filter on the molecular consequences. I can filter on variant type. In this case, they're all, and in the case of human, they're all SNPs, I can tell you. And in the case of human, they're all variants, and we'll talk about what else that could be in a minute. We have not loaded any disease or phenotype annotations for the human variants into the Alliance yet. That it's on the list of things to do, but we haven't had a chance to do that yet. So these columns for has disease annotation, has phenotype annotation, these are all blank for human, but at some point in hopefully the not too distant future, we want to start importing disease annotations into the Alliance like we do into RGD, both from ClinVar and from other sources that Ensembl uses, where they've assigned disease annotations directly to variants. So that's coming. It's not there yet. Anyways, if I choose a filter, so I choose missense variants, then I've reduced my number from 47,000 down to 948, which is a bit more of a manageable number. And if I want to look at more information about a particular variant, the Alliance runs a pipeline for all of the variants, and that pipeline returns transcript-level consequences for all of the transcripts of the gene, and that comes from the Ensembl Variant Effect Predictor. So all of the variants are run through the VEP to get additional details about the variants, and that's shown on the variant detail page. So variant details for Lepr, you've got just your, the name of your variant and all of the information that appeared in the summary table on the previous page. But in addition, you have the list of, they're called sequence features, but in this case, they're all transcripts. So transcripts both from Ensembl and from RefSeq for the Lepr gene. It tells you whether that transcript is protein coding or not. It tells you what gene it's associated with. And you'll notice that in this case, because Lepr and Lepr are overlapping, some of the variants would be assigned to transcripts from both genes. So this is actually, these are all the same one variant, and then there's information for that one variant for multiple different transcripts, and 4 of those transcripts are for the Lepr gene, 1 is for the Lepr gene, and there are actually quite a few more rows for this particular variant. But as you scroll farther to the right, there's additional information such as the variant location, whether it's in an intron or exon, and which intron or exon it's in, the molecular consequences such as start lost or a missense variant, what VEP predicts as the impact of that variant for that particular transcript. So for 3 of these, they're high. For 2 of these, they're moderate. And then there are predictions from both SIFT and polyphen for whether the variant is damaging to a particular gene product or not. And in this case, I have actually filtered on the SIFT prediction so that I'm only showing deleterious variants. And then you've got the SIFT score, and then you can see that SIFT predicts all of these to be deleterious, but polyphen is predicting 3 of them to be benign and 2 of them to be probably damaging. So you can see that there's differences between the predictions from SIFT and polyphen, and that might be of interest. So looking at non-human species, in this case, I'm looking at rat. For non-human species, both the variants that are associated with phenotypic alleles and high-throughput variants have been loaded. So the human is only variants that are coming from databases like ClinVar and also from like the 10,000 genomes, human 10,000 genomes project. So mostly for whole genome sequencing. For non-human species, we have manually curated variants that are associated with the phenotypic alleles, in addition to high throughput variants. And the sequence viewer, there's a sequence viewer that shows on the gene page that gives you the transcripts of the gene, and the sequence viewer shows only the phenotypic allele associated variants in the context of all of these transcripts of the gene. Because of there's so many high throughput variants, if you put the high throughput variants on in this display, it would basically just be a line across it. There would be very few breaks. So at this point, only the allele associated variants are showing, but you can see that in this case, in the case of rat Lepr, there are 2 allele associated variants that have been curated and are showing on this display. And here you can see these are the 2 variants right here, the 2 variants that were shown above. But there are additional alleles that don't have variants curated for them, either because they're not known or because we haven't gotten to them yet. So here is the where there are additional categories of allele variant data. So these are all alleles that don't have variants annotated to them. These 2 are alleles that each have 1 associated variant, and for some of the species, there are alleles that have more than 1 associated variant. So it would say allele with 2 associated variants and show both of the variants, for instance. And then you've got the high throughput variants that are categorized as variants. And you've got the HGVS nomenclature is used for both the symbol and for the variant designation. This link will go to JBrowse at the Alliance so that you can view this variant in its genomic context. Before I talk about the variant detail page, I want to point out that some of these alleles and both of the alleles that have variants associated with them have disease annotations. And this one, the Lepr FA, also has phenotype annotations, as do some of these other alleles. And so if the allele has either disease or phenotype or both annotations, that will be shown in these columns, and then these will link to the actual disease or phenotype annotations on the allele report page. And we're going to look at the allele report page in just a minute. So when we're looking at alleles, if the allele like this one has a variant associated with it, when you go to that allele page, so this link takes you to the allele page, it will show you details about the genomic variant that is associated with that allele. And you can see that there's I'm running out of time. Okay, lots of information. Variant molecular consequences have been predicted. So we've got the amino acid change, you've got the codon, and then the amino acid for both the reference and the variant. The allele records contain disease and phenotype annotations where they're available. And variants not associated with alleles have variant record pages. There is no allele page, obviously, but there is a variant record page. And on that variant record page, because you're only looking at the information for a single variant, you see that variant in the sequence viewer. So here you can see that the variant is at the far 3' end of the gene. And again, you have the consequences displayed for each of the transcripts that are associated with that gene that the variant overlaps. So one type of data about rat variants that the Alliance hasn't loaded yet is strain assignments for variants that are derived from whole genome sequencing. And I'm not going to have really time to go through this in detail, but I will mention that our variant visualizer is a search engine to find rat strain, human ClinVar variants, rat strain variants across 4 assemblies, ClinVar variants across 2 assemblies, and we're in the process of loading GWAS variants into this as well, and then variants from the Dog 1000 Genomes project. And so you select strains or genes. Here I've selected 3 strains, put in a list of genes, and I didn't show, there's another page that allows you to choose things like whether the variant is damaging. Here I've selected either possibly or probably damaging polyphen prediction, and I get the list of these genes that I've put in and how many variants match my search criteria in each of these strains. So in this case, there's variants in LH and not in LL or LN. LH is the hypertensive of the 3 rats. So if I click on this box, it shows me what those actual 2 variants are. And if I click on one of these boxes, I get a variant detail page that gives me information about the variant and about the sequencing. It shows me the reference and variant nucleotide, the reference and variant amino acid, and then shows me that in the context of the full protein sequence and gives me information about how polyphen is predicting whether this is damaging versus this this particular transcript. And I can tell you that the other transcripts that it's not predicted to be damaging for the other transcripts of this gene, just for this one. And with that, I'm going to say thank you. And we've got maybe 2 minutes for questions.
Great, thank you so much, Jennifer. So much work going into these databases, and I think just really helpful for our curation work. Thinking, for example, about gene disease clinical validity and rat models, and how we can use that information in our curation. So thank you for all of this information. Does anyone have questions? I do have a quick question. I think a little while back you mentioned when a gene is labeled as inferred from expression pattern. So this would presumably just be something like a candidate gene, like if it was expressed in muscle, for example, it would be a candidate for a muscle disease. Is that kind of how would that work?
Very often, it's not 100% true. Sometimes they will look across different tissues, for instance, and find that expression has changed in a tissue that you wouldn't necessarily expect to be associated with the gene with the disease that they're looking at. So, maybe you see increased expression in the brain, but it's associated with a muscle disease. And so it's not necessarily clear why that would be the case, although the researchers might have said something, but we would annotate that even though we're not quite sure why it would be different in brain if it's a muscle disease. But I will tell you that we're pretty stringent in our criteria for when we will make, especially the IEP annotations. It has to be statistically significant. They have to actually say this is statistically significant, and preferably give us the p-value so that we can actually see that the p-values are significant. We don't just, just because something might have looked a little bit different on a graph, we don't make an annotation based on that. It has to have significance.
Okay. I'm just curious, how many curators do you have